recorded, and a link to the meeting recording will be available at the end of this meeting and shared with all participants. So today's webinar is focused on a recently funded project that is exploring the potential benefits of Florida seaweed for Florida cattle applications. We're going to provide a broad spectrum of information today. So you're going to see a variety of links and a variety of resources that are references. Please note that we're going to record the whole thing. You can sit back and relax, and we will provide a PDF to everybody that's registered with all of the relevant resources and references and links after this meeting today. So go ahead and just relax and pay attention. Again, we note that the meeting is being recorded. If you have a question along the way, please feel free to just type it into the chat, and one of us can probably get to it right after you think of the question. But there will also be an opportunity for you to ask the speakers questions directly at the end of the meeting, and you can kind of pick their brains verbally if you don't want to type your question in the chat. I'm going to take just a moment to introduce our team. My name is Angela Collins. I work at the Tropical Aquaculture Lab, and I'm just sort of facilitating the brain power that is this team that's on this meeting today. So I'd like to introduce Dr. Nico Di Lorenzo, Dr. Lisa Garcia, and Dr. Jose Dubois. These folks are leading the charge on finding applications for seaweed in Florida cattle systems. They're located at the UF IFAS North Florida Research and Education Center up in Mariana. Dr. Aaron Welch is also joining us today. He is our industry partner and the owner and operator of Two Docks Shellfish. He and his team are providing fearless seaweed farming expertise and an industry perspective from the shellfish aquaculture industry. Both Aaron and I are in the Tampa Bay region, but I'd like to note the network of other colleagues that are informing this research that are also at the University of Florida, and we're going to hear a little bit about some of their work also today that are directly informing seaweed research here in the state and the different applications that this product might have for our aquaculture industry. Dr. Ashley Smith, Dr. Andrew Ropicki, Gabby Forza, who's our PhD student, and she's here with us today, and Dr. Dale Laughinghouse. All of these team members have done work looking at different seaweed applications here in Florida and are just starting to investigate some of the potential for this industry. So I wanted to give you just a tiny little bit of background to start this conversation off. At a global scale, seaweed aquaculture is one of the fastest growing sectors. In 2021, the value of seaweed was estimated to be almost $16 billion. There are over 12,000 different species of seaweed in the world. Only maybe like 25 of those are actually produced commercially. But markets for seaweed include everything from food and food products to things like personal care and health products, animal feed, biopackaging. There are also ecosystem services that are celebrated, and different avenues for macroalgae have been considered as ways to restore water quality and provide mitigation markets. However, it's important to note that Asia currently dominates over 99% of the global market, right? So in the United States, seaweed aquaculture is very much still an emerging industry. Even in states where we do have production and is considered high production in the US, the industry is still kind of considered in its developmental stages here in the US. And in Florida, we have very limited, almost zero seaweed species that are being commercially produced, and those that are at a small scale for like the aquarium trade. So the current markets that exist for seaweed are pretty volume driven. An expansion of this product in the US would depend not only on a viable market, but also on adequate infrastructure and processing and packaging facilities to deal with the product that might be produced. So in Florida, the conversation is kind of just starting, right? And at first glance, Florida seems like a prime candidate for producing seaweed. We have lots of water, lots of coastline. We have a plethora of different seaweed species, as you all know, if you walk along the beach. We have existing working waterfronts and an established fleet of working watermen and women in our shellfish aquaculture community, as well as our commercial fishing industries, right? But we have a lot to learn very to begin with, like what species here are actually viable to be produced at scale and consistent for a market. What will the regulatory framework look like? How much will it take to enter an existing market or develop a new one with the structures that we have in place in Florida right now? So this kind of just leads me into this flurry of activity that's been surrounding seaweed research for the past few years. A lot of those projects are being led by folks at the University of Florida, and working directly with our industry partner in Two Docks Shellfish Company, who was one of the first industry members in the state to explore this potential avenue from a research perspective. So we're going to dive into a little bit of some of that today. And we're going to specifically focus on implications for cattle systems here in the state, which is kind of what this particular project that we're discussing today is all about. I just wanted to go ahead and give you a quick summary of the seaweed, current status of seaweed, not only globally, but also here in Florida. And today's all about managing expectations too. We're not at all saying that the seaweed market exists currently. What we're saying is we're exploring and gathering data to inform the potential of seaweed aquaculture for all different applications within Florida's agricultural community. So without further ado, I'm going to go ahead and pass the microphone to Dr. DiLorenzo, who's going to give us a little bit of an overview of his current project.
Perfect. Well, thank you very much. Thanks a lot for the introduction. And thanks, Angela, for all the work you've been doing over the years since, well, our short history working together to convey all the messages of all the research we're doing. I think you emphasized quite a bit how preliminary some of this is, but also it's exciting to see some of the figures that you show about the growth of this segment. It's totally new to me, and I won't take too much time introducing myself, but I will say, just for the sake of this brief presentation, first I want to have a discussion, especially I think at the end we're planning for the questions. I really would like to hear from experts on the field on the potential of this project, because my training is as a ruminant nutritionist, beef cattle nutritionist. I'm located, as Angela said, in Panhandle of Florida and Mariana. Same with Jose and Lisa. We have a REC, North Florida Research and Education Center, where all the three of us work in the beef and forage team. And this is totally a new area for us. We're very curious, and we're fortunate to be funded on this Sea Grant project that got us started. One of the main things, regardless of how successful we are, to me, already the huge success of this project is the ability to put us in contact with people like Angela and her team. Definitely Aaron and his team has been a tremendous opportunity to go to the Tampa Bay area and learn what they're doing. I think it's going to take projects like this, interdisciplinary thinking outside of the box, to solve some of the huge challenges that we have in the state. So how to balance food production and urban encroachment, and how we're going to get to that, balancing resources and environment. So I think that's where all of the things converge in this project, and I think we're just scratching the surface of the potential of this. So again, my presentation comes from the perspective of a ruminant nutritionist, so I have probably a ton of questions for my colleagues on the Sea Grant team and people working in the different areas related to aquaculture, because I really think it's something that we're going to see a lot of growth here. I hope we see a lot of growth in Florida. So again, thanks, Angela. I want to thank the entire team, Aaron and Andre Brugger, who also was instrumental, were two of them in the development of the proposal. Without Aaron, there's no way we could have done any of this. So huge thanks to him. And I know he put a lot of human capital into this. We have a hurricane in between and everything else. So yeah, this is also a small story of resilience on this team too. But I without any further ado, I'm going to get into some of the topics of why we're talking about this and what do we have in Florida going for us. So there's a lot of challenges, as you probably hear in all the news how rapid Florida is growing. It's a third state in the union in terms of growing population. And we're not certainly as big as California or Texas, so we got a lot of people very close to each other, and we got a lot of coastal area. We'll talk about that in my next slide. But the fact that it's growing about you hear it in different ways put together, but some people would say this a city the size of Orlando every year, or about 900 new residents per day. It's really scary when you see some of the numbers. This creates a lot of pressure on agriculture and aquaculture. Oops, sorry, this might be on a timer because it moved before. So aquaculture and it's one of the things that we're going to be talking a lot today. But there are also other industries like the industry of the sugarcane industry that produces a lot of molasses, citrus pulp as a byproduct, huge cotton and peanut industry in the Panhandle. We have a lot of byproducts. So there are a lot of opportunities for this circular bioeconomies, as some people would define them. And related to that, one of the things that we have really going for us in Florida is the coastal zone. So if I'm correct, the Florida is the second state, only second to Alaska in terms of the length of the coastal line, and obviously huge difference in terms of climate. And so the opportunity to do this type of synergistic projects, interacting with our friends in aquaculture, it's unique to Florida, and there's plenty of opportunities for that when we look at the length of the coastal. If you think about that as a potential area where we could harvest, in this case, something that it might fit into the cattle industry and at the same time create a bioeconomy, the area of that, it's definitely one of the opportunities, because we have against us the land area, which despite that land area, we still have a lot of cattle in that. I'll show you here in a minute. But if there's a way that we can really expand, will be into the water. So the cattle, like I say, it is, if you're not familiar with it, and I do recognize some of the people in this seminar, the webinar, they're quite involved with the cattle industry. They probably also know a lot about the coastal areas, but we do have, when we put it all together, like beef, cows and calves, bulls, everything, about close to 1.7 million head of cattle, close to 900,000 head of beef cows. And that is a huge economic sector within the agriculture. Cattle has been one of the strongest segments in Florida. We do have the challenge of many Florida pastures are low in protein, but they're very high in production. And as my colleague here, Jose Dubé, is doing a lot of work in that area, and there's a whole team of people in Gainesville working on forage breeding. Sorry, this is on a timer, so it's going to keep changing. So I'll keep pushing it back to the correct slide. So it would be, we're hoping to always come up with forages that are higher in protein, and there's a lot of effort into that. But at the same time, there's a limitation on what we can provide in terms of quality of those forages, even though the quantity, it's usually not a problem. But there is a need to develop local and high protein sources, because even as times of the year like right now, we have projects going on here in the Panhandle where we are feeding some of our common supplemental forages like Hermathria, Limpograss, but we do need protein to supplement that. Otherwise, cattle cannot make it without. So the need to reduce the reliance that we have from external protein sources. External, and when I say external, I'm talking about the Midwest typically. So most of our protein sources, soybean based, come from the Corn Belt area. So there is an opportunity to develop some local sources. Today, if you're not familiar with the cattle industry, the only protein source that is somewhat locally produced is cottonseed meal. Used to be some of the distiller's grain, another great byproduct from corn used in the ethanol production, but there are not very many plants left in the region. So really, right now we're down to only one product, and that's where this project could have a tremendous impact. So I know it's a big jump. So from there, we're jumping into something that you see in that slide that is bromoform. But let me do a very quick introduction of that so that you get to do a little smoother transition. So seaweed as a feed additive is something that started maybe 10 years ago, but it wasn't until really 6 years ago that we started seeing some of the first papers on this. And there are several ways in which seaweed, as a very broad term, maybe some of my colleagues in aquaculture they think that this could be defined better, but seaweed may be a broad term for a lot of the things that we're trying to look at. But there is some potential for seaweed to enter into cattle feeding, not only from the protein standpoint that I just talked about earlier, but also from the standpoint of mitigating enteric methane emissions, which has been it's a topic that it's pretty hot still in most circles. We're still talking about how to reduce the carbon footprint without giving up on efficiency, because that's the number one way we're going to do that, increasing feed efficiency. But at the same time, there are opportunities to reduce the carbon footprint by creating additives, and that's why bromoform is here in the picture. Additives that have the capacity to reduce methane. I will talk quite a bit more on the second part of my presentation later today. I'll talk more about bromoform and why we talk about bromoform, seaweed, and methane and cattle all in the same sentence. So just bear with me, and we're going to come back to that. But for now, for this part of the presentation, I just want to introduce the concept that some of the microalgae that we've been playing with in this project have bromoform, and bromoform is a key compound in reducing enteric methane. So we'll come back to that. So overall, this project, the main goal of this project was to evaluate Florida grown, and I will emphasize the Florida grown part of it because that's really what's unique about this project. Florida grown macroalgae as livestock feed with the potential to reduce methane, but also with the potential, as I'd say in that third bullet, to develop or to have a high protein or introduce high protein into cattle diets, recognizing that that's one of our challenge. And those two together, that's why I talk about the first and third bullet, really combine to develop the secondary goal, which it is to develop a sustainable coastal aquaculture industry. And I know there is one already more related to maybe human food and other uses, but I really do see a tremendous opportunity to develop an aquaculture industry and start talking about this famous circular bioeconomy that I hear so much about. I truly think it's quite attainable. I think there is a lot of potential, but I didn't realize how complex it was until we started this project, and especially when it comes to scaling up some of this production, as Aaron may address later today, or at least mention. Like I say, we could not do this project without him and his team, but also you have to be aware of the tremendous logistical challenges that are involved with producing a product that cattle will have to consume to the tune of 20 pounds a day. So it's unfeasible to produce the amounts that we may need. So we'll have to be very strategic, and that's where the last part of my presentation will come about. How can we, in a realistic way, produce microalgae or harvest microalgae in the coastal regions and still have an impact on the cattle production? So, for the phase one of this project, our first approach was to harvest algae in the wild. Basically, you see on the top picture Georgia Dubé working as a PhD student. She did a master's actually also working with algae and in vitro system. We had some really neat results that were actually part of the other grant that Angela mentioned earlier, the one that Ashley is the PI. So we also explored the potential of sargassum, recognizing the huge problem that it is sargassum to our
No, qué bueno.
coastal areas. We tried to use that. We had our own challenges with that I can address in the questions section. But Georgia there is separating or trying to actually collect a subsample of some of the microalgae that we were harvesting at that time. So the project started with wild harvesting of microalgae, getting on a boat, Lisa and Georgia both driving to Tampa and several occasions, meeting with Aaron and his team, and then getting on a boat and harvesting all this algae, which later on we evolved towards a more controlled situation. It's sort of what you see in the picture on the bottom. That was a part of our visit to the facilities at Two Docks Shellfish. When we went to Two Docks, we got to see some of the tank where now we are growing some of the algae, and that's a picture that you see there. But the project started with wild harvest of this algae and identifying what are some of the best candidates. So we require a lot of help from many of you in this webinar to identify species and to learn a little bit more about the biology of that. So it's been a great learning process. So let me briefly tell you about this, the phase 2 of this project, and this is my last time before we switch gears and we cover some other aspects, and I'll come back to a more in-depth explanation of the project. But in a nutshell, everything that you see here is, I'll try to explain it as thorough as possible. So we have certain animals that you can see. I think you can see my pointer, well, the arrow here. It might be, I don't know if I have the, like, I'll use this instead of the pointer. So we have cannulated animals. You can see here. These animals, if you're not familiar, they have a sort of a window or a hole in the side, which allows us to collect what we call digesta, which is ruminal fluid, where all the microbes that are part of the digestion of feed. We collected that, we transported to the lab in thermoses to maintain the anaerobiosis, because this is a highly anaerobic system. In the lab, we do a certain thing. We mix it with buffer to simulate saliva, and we conduct incubations at 39 degrees Celsius with carbon dioxide, basically simulating in each one of these flasks. Now we're simulating a cow. We typically use this bottle that you see here. We call them serum bottles. So then each bottle is truly a cow from a fermentation standpoint. We incubate for either 24 or 48 hours, and then we measure total gas production. Why we do that? Because in order to assess methane, we need to have two components: concentration of methane in a subsample of gas and total amount of gas produced. We also do some other things like volatile fatty acids that you see here and ammonium nitrate. Those are other parameters resulting from the fermentation of feed by cattle. And we measure those because it provides a lot of information about what we're doing. But then a subset of those samples, we perform what you see here as IVOMD, which stands for in vitro organic matter digestibility. Again, that simulates a cow, each one of those tubes, and after a period of 24 to 48 hours, we add pepsin hydrochloride to simulate some other parts of the digestive process in cattle, and at the end, we evaluate how much of that is digested. Why we do that? Because at the same time that we're attempting to measure or to mitigate methane, we also want to make sure that we don't hurt digestibility as a proxy for the productive performance of the algae and the cattle and the feed in general. So we want to make sure that if we develop something in the future, it's something that is not going to compromise production, because as I say earlier, that's one of the most important legs of the sustainable growth in terms of beef production. It has to be sustainable from many aspects and not only the environmental one. So that's kind of what we're after. And here on the right, you see on the right part of this picture, you see actually three pieces of technology that we used a lot. They're quite expensive, but that they complement our research greatly. At NFREC, North Florida Research and Education, we have something called a green feed system. We now have three of this. This unit basically allows cattle to approach that machine. It trickles, it gives them a treat. For that, we use typically gluten. And while they're eating that treat, they're breathing into the machine. And this machine, there's a tube here that you can probably see it on the screen, a flow meter here, the little green box, and there's some elements in the bottom that basically are sensors for methane. And what we get is methane emissions from that machine. So cattle breathe into that machine, and on a real time, this is plugged into electricity. So we're getting actually the most important part. There is a modem down here that sends a signal to a cloud, so it collects methane data. Cattle wear a radio frequency ID tag on their left ear, so when they enter the machine, we know exactly what animal it is and what treatment that animal was on. And the treatments and some other projects, and likely when we get to the volume of algae that we need, we're going to use this red machine here on the left, which is called Super Smart Feed. That's just a fancy name for a precision feeding tool that allows us, again, based on radio frequency ID of this cattle, each one can eat out of 4 bins. They're based on their RFID, and depending on which bin they're eating from is the treatment that they get. So basically, we have all the animals confined in one big pasture, actually more of a dry lot, because you see there's not much here to graze, and that's by design. They get all their feed in this feed bank that's on load cells. So basically, between these three components, so this feed bank, the green feed, and the super smart feed, we basically can control how much each animal eats and out of which treatment. So that's a way we're planning on moving on the next phase of this project when we move into an in vivo. These are some of the tools that we probably be using. So I just wanted to introduce you to them. So with that, I'm gonna stop at this point, stop sharing, and turn it back to Angela for another segment of this, and I'll come back to talk more in details about the next phases of the project.
Thank you. That was fantastic. I'd love for any of our listeners, if you have any questions along the way, don't forget that you can type those into the chat. As Nico just gave a really good overview of their particular research project right now, and one of the foundations for that work actually started with the UF IFAS Seeded project that both Nico and Jose and Lisa and Aaron were involved in, and Ashley Smith was the PI on that one. And so she unfortunately is not able to join us today, but she was able to record a little bit of a presentation that I would like to share with you. So I'm going to go ahead and share that. We can listen to that for about 10 minutes. Gabby is here, and Gabby is our PhD student who's been involved in all of these different seaweed projects. And so she can also answer questions related to this. So I'll go ahead and share that. If you guys have any trouble hearing it, just let me know. I'm going to go ahead and do that.
Good afternoon, everyone. Hi, I'm Ashley Smith. I'm an associate professor at the University of Florida's Tropical Research and Education Center in Homestead, and I'm based in the Soil, Water, and Ecosystem Sciences Department. I'm sorry that I can't be there live with you all today, but I'm thrilled that I can still share with you some of our work on seaweed aquaculture in Florida and talk about how it's evolved from this pilot project into 2 major research initiatives. When I think about our seaweed project, I think of it less as a story of seaweed, although some might get angry when I say that. And I think of it instead as turning a shellfish industry nuisance into an opportunity for economic diversification, water quality improvement, and climate-smart agriculture. And I'll spend a few minutes today talking about the foundation that we built from our Seed It project that led to the projects that are being discussed in more detail today and some of the other projects that we have. So why seaweed? Why now? First, Florida's shellfish aquaculture industry is looking to diversify. Oftentimes, they're closed for harvest from things like red tide, and seaweed could be another market that they could be involved in to help offset some of those costs. Seaweed also provides environmental benefits, such as nitrogen removal through bioextraction. This is essentially pulling nutrients out of water, and this has water quality implications. Florida also has warm waters, which would allow for year-round cultivation of tropical species, which is very different from what we know about seaweed farms up north, where you see like kelp. And there's just this general interest in seaweed on a global scale, where we're seeing seaweed in more products, from food to fertilizer to plastics, that there's just this increase in demand. And so it's this increase in demand and interest and all of these reasons that we wanted to see if seaweed was a viable opportunity for Florida. And to do that, we wrote a grant. So a bunch of us got together and applied to the UF IFAS Seed It program. Seed It stands for Support for Emerging Enterprise Development Integration Teams. I know, it's a mouthful, but the Seed It program is really designed to find new products, create new partnerships, and support Florida's agriculture and aquaculture production. And our project brought two industries together. It brought the seafood industry and the cattle industry. And it also gave us the chance to explore ways to improve our environment. I've talked a little bit about why the shellfish industry might want to be involved in seaweed, but we also had the cattle industry. And the cattle industry was looking for ways to reduce their carbon footprint. And that's really where these two industries came together. A study from Australia found that certain types of seaweed had a special compound called bromoform. And that compound, when fed to cows, would make them produce less methane. So it's this desire to produce less methane and lower the carbon footprint from cattle, while also diversifying opportunities for this aquaculture industry that led to our project. And our project had 4 key questions. We wanted to look at the nitrogen removal capacity from seaweed aquaculture. We wanted to see if our seaweeds had bromoform in them. We wanted to actually look at the economics of producing seaweed here in Florida, and then see what people were willing to pay for climate smart cattle that might have been fed seaweed and therefore had a lower carbon footprint. So I just want to share with you some of the key results from our Seed It project. The first was that we can grow seaweed. Dr. Erin Welsh at Two Docks Aqua Farms was able to produce Gracilaria. And Gracilaria is a brown macroalgae that has the potential to contain bromoform. So that was huge. Now that we were able to grow seaweed, we could ask some of our other questions. The first was, how much does it cost to produce seaweed in Florida? And we found out that one seaweed lease can produce 3 tons of Gracilaria per year, and that the cost of production is about $7 per kilogram. Dr. Andrew Rapicki, who you'll hear from a little bit later today, can go into the details of this because he was leading that part of our study. We also then figured out, does the seaweed that we can grow here contain this bromoform? And yes, it does, but at low concentrations. And we identified that one of the bigger challenges was related to getting the seaweed from the leases to the lab without degrading the bromoform. We also then were able to ask how much consumers were willing to pay for the ground beef with a lower carbon footprint. And we found out that consumers were willing to pay a substantial premium for seaweed-fed beef that had a lower carbon footprint. In fact, they were willing to pay about $1.40 to $1.80 more per pound, but only when they were explained how the benefits worked in plain language. So the marketing really mattered here. I learned a lot working on this project and had a lot of fun doing it, and the project team has a lot to be proud of. Aside from new collaborations, we also cultivated new relationships with Atlantic Sapphire and Natuna, 2 companies interested in seaweed aquaculture. And we were able to work directly with the industry to support them to develop and pilot tests and protocols to lay the foundation for new projects and products to come. We also created new collaborations between the cattle industry and Florida Sea Grant, groups that had never really worked together before. We quantified nitrogen removal benefits. like being able to link the seaweed to water quality goals. And we were able to support 3 graduate students. In fact, here is one of them presenting a poster at a conference. And perhaps the most important part of this project for me was that we built capacity for our aquaculture industry partners to begin to develop new products and to investigate new revenue streams. There was a lot of interest generated from our CDIP project and a lot of new questions. So we were actually able to get 2 new projects funded. The first was from Florida Sea Grant, which focused on cultivation of red macroalgae for cattle feed, which you're going to hear a lot more about today. And the other one was from the National Sea Grant Office, as one that I'm leading. Our project builds on the CDIP project to identify different seaweed species that naturally occur here in Florida and their potential for commercialization. We're also going to be developing protocols that help scale up from the lab to the field and assessing the market potential and feasibility of having seaweed aquaculture here in Florida. So ultimately, our central question is really seeing if seaweed is a good fit for Florida. And to do that, we're going to characterize the different species that occur naturally at our shellfish leases, including their traits and potential uses. We're also going to quantify nitrogen uptake data, which is what you need to see if seaweed could be used in water quality restoration. We're going to be developing these cultivation protocols for the lab and land-based systems to help understand how to optimize conditions for tropical seaweed species that would lead to production of more profitable compounds or species. And then we're also going to really look at the market to see what the potential applications are for the seaweed species that we can grow here. Whether that's food, cosmetics, or fertilizer, we need to see what's out there and what the options are for the seaweeds that we can produce. And then we're also going to build some tools that we can use to assess the feasibility, costs, and scalability of seaweed aquaculture here in Florida. And ultimately, what we'll do is produce outreach material like webinars like this and other workshops to share our findings and make sure that they're accessible. Because we want to make sure that our work goes into making informed decisions about whether seaweed becomes a new product for Florida or not. And that's okay too. As you can see here in this photo, this is Gabby. She's a PhD student on the project right now, and she's working diligently to process all the seaweed that she's been collecting. The work that we're all doing for this project is meticulous, and it is the groundwork that we really need to start to make recommendations about the feasibility of seaweed in Florida. So where do we go from here? Right now, our project is actively underway, and this is a photo from a recent field trip that Gabby went out and collected some seaweed from Aaron's lease. And we're looking at that seaweed, we're characterizing it, we're seeing what seaweed species are there, and then we're doing some lab trials too to start to figure out how to grow the seaweed and what conditions they might need to best grow and produce some of these potential high-value compounds. So there's a lot more that our group will be working on in the next few years, so stay tuned. You're not done hearing from us. And with that, I just want to say thank you. Thank you all for your time and for all of our industry partners who've been helping along the way. And I'm really sorry that I'm not there today to talk more, but Gabby is there, and I'm sure she'll be happy to answer any questions.
Okay, that was Ashley. And again, I don't think I mentioned it. Ashley's based at the IFAS Tropical Research and Education Center down in Homestead. And what is so cool about this project, all this conversation today, is just like the cross-cutting disciplinary collaborations, right? We have the cattle industry, and we have animal science folks, and we have agronomy, and we have psychology, and we have Florida Sea Grant, and we have biogeochemistry, and we have marine economics. We've got Andrew Ropicki here with us today from the Food and Resource Economics Department, and he's going to talk a little bit about some of the potential for a market for seaweed in Florida based on some of this work that's been established. So Andrew, I'll go ahead and toss the mic to you.
So is it showing my slides now? Okay,
yes,
perfect.
it is. Perfect.
So yeah, Ashley's presentation kind of touched on some of this a little bit, but we're going to go into more detail. And fair warning, the willingness to pay numbers you're going to see are a little different because we're kind of showing you more of an apples to apples comparison when you see them. So the willingness to pay values will be a little different, but we'll kind of get into that as we go. So this was work we did in the Food and Resource Economics department as part of that initial Seed It project. And the consumer preference work, the willingness to pay was really spearheaded by my colleagues, John Lai and Bashir Cassis. And their wonderful graduate student, now postdoc, Zhang Wei, did a ton of the work, did an amazing job on this. So jumping right into it, we did a big national survey online using Qualtrics. This was May to July 2024. For our willingness to pay part, we had 962 responses we used. Actually, we had over 1,200 where we gathered just general consumer sentiment and preferences and beliefs regarding seaweed fed beef. The data gathered that was really important to the willingness to pay was grocery shopping behavior, preferences for ground beef, things like fat content, all the way back to the basics. Do you even eat ground beef? Things like that. We got some general kind of baseline data on their environmental attitudes and concerns. Then we did the willingness to pay for seaweed-fed ground beef. This is what we call the discrete choice experiment, which I'll discuss a little bit more in just a second. And then we gathered demographic information just to make sure we had a representative sample of U.S. consumers, which we did. And Ashley touched on this a little bit. One of the neat things we did was what we call our information treatment. We tested how information, how it was presented, impacted willingness to pay. And we kind of looked at kind of using scientific language versus kind of common language to see how it changed willingness to pay. And we got some interesting results there. We'll get to in a second. Okay, so a choice experiment is a tool economists use to value goods and attributes of goods. And the way it works is, and this is an actual choice that could pop up in the survey we did, where you give a consumer, you say, here are your choices, which one of these would you buy? And you can see we have 3 different types of ground beef, and then we've got a control. If they don't want any of them, they can select, I would not buy any of these. And what we vary among these choices are the price of the ground beef, the feeding method. We had 4 different feeding methods. We had grain-fed and grass-fed. Then we had grass-fed plus seaweed, or seaweed plus grass-fed, and seaweed plus grain-fed. We varied the fat content because, as everyone knows, that's a big thing when you're buying ground beef. And then some of them we had an organic label versus no organic label. And each respondent to our survey saw 8 of these choice sets. And from that, it varies, it randomizes. We get enough information, we vary those variables through those 8 choices, and we get enough responses, we can kind of get a good handle on how people value the different attributes of the ground beef. And our focus here was really on the seaweed diet supplementation. So our information treatment, I won't go into too much detail for time, but we really had 3. Our control is we don't tell you anything about feeding seaweed to cattle. We just jump right into those willingness to pay questions and see what your willingness to pay is. Some people got that. Others got the scientific language, which, was a little more technical regarding the environmental benefits of seaweed supplementation and cattle feed. And then lastly, we tested, some people saw a kind of a common language or layperson's language treatment, which was designed to be a little more approachable, a little less scientific-y in nature. So those were our 3 treatments. So kind of jumping to the results now, what you're seeing here is we've got rows for each of our treatment groups, our control, scientific language and layperson's language. And then, like I said, you're seeing different numbers here. And what you're seeing is we kind of wanted apples to apples comparison. So we did grain fed supplemented with seaweed compared to grain fed, just plain grain fed, and then seaweed plus grass fed compared to just grass fed. And the first thing you'll notice is that the control, when we provide no information, willingness to pay is actually negative. Consumers did not, on average, or respondents to our survey, did not, on average, have any understanding of the environmental benefits associated with this, the idea of lower carbon emissions. And as such, they seem to think, why would I want you to feed seaweed to cows? With the scientific language, we presented them with the information that cattle production leads to carbon emissions, and this seaweed supplementation could lower those carbon emissions. And with that information, willingness to pay became positive. You can see for the grain fed, the average respondent was willing to pay 50 cents per pound more. For the grass fed, it was 28 cents per pound more. And then what was really interesting was the layperson's language. We got bigger bumps. So that was kind of interesting. Apparently the message kind of hit home a little more with the kind of simpler language. And this is kind of interesting because this might have implications for other goods and services where either we're talking about a potential negative environmental aspect that can be mitigated in some way, or similar to what we're talking about here with seaweed supplementation. But the messaging matters, how you get that information across, and we found that interesting. So kind of key takeaways, consumers would need information on the environmental benefits. It's not common knowledge, or at least it wasn't in 2024 when we did this survey. And without it, willingness to pay was actually negative. But when you explain the benefits, willingness to pay becomes positive. So there is a premium that seaweed fed beef could capture. And then using plain language, as we said, led to higher willingness to pay than the scientific language. Potential additional research in this area that would really be interesting and be fun to do. So we focused on the benefits of actually feeding the seaweed to the cattle, kind of that aspect of it. But there are benefits for growing the seaweed, if you think about it. As you're growing the seaweed, it's removing carbon, nitrogen, and phosphorus from the water. So it's actually cleaning the water. There might be, with that information, you might get a higher willingness to pay for this product. And one thing we were really kind of interested in and would like to look at is, does that willingness to pay vary by location? Really thinking about Florida, a state where, we associate our state with our coastline and our marine environments, and water quality is so important. With knowing that this seaweed was growing here and is helping to improve water quality in coastal areas, increase that willingness to pay more. That'd be something fun to look at. Looking at willingness to pay for additional types of beef and milk would be interesting. Maybe there's a higher, ground beef is a very common way beef is consumed, but maybe steaks, the higher end products, maybe there's a higher willingness to pay. That'd be interesting to look at. And then lastly, looking at the potential for increased consumption of beef. I mean, there are consumers out there who no longer consume beef or do it in lower quantities than they maybe previously did because of those negative environmental consequences of raising cattle. So if we can mitigate those, maybe you bring consumers back to beef or, new consumers. So that's some of the stuff we'd also like to look at. So this was a really quick overview. Since we're recording this, you can look at this stuff. If you want to dive in a little more on the willingness to pay stuff, John Way, we have a manuscript under review, but his dissertation, that chapter is publicly available. So, if you need more than just I can do in 10 minutes, we can do that. And then that we also have a publication out there that kind of deals with more general stuff on consumers' perceptions and attitudes from the survey itself. And I think, yeah, that's it for me. So I'll stop sharing.
Okay, wonderful. Thank you. Thank you, Andrew, for that summary. And before we move into the next presentation, I see that Emily's got her hand up. Emily, I'm going to ask you to keep it really short or save it for the end, but I'll go ahead and unmute you if you would like and ask your question if it's short.
Yeah, I can ask it now or later. Let me know, Andrew, what's best. But I was just curious when you showed that matrix of your conditions, why it seemed that they would pay less of a premium for seaweed and grass rather than seaweed and grain, because I would have assumed like the seaweed and the grass were both a premium, but you didn't actually see that reflected. So I was curious if you had results for that or any ideas, and feel free to touch on this later if it's best for time.
I can do it really quick. You completely randomize those, so some of them will not make any sense to you. What we would think is that people would pay a premium or would expect to pay a premium, at least for grass fed, but we want to know how their demand for beef. So we need to know if they're the person who doesn't want grass fed under any circumstances, even if it is cheaper. So they see 8 choice sets. It varies through them, and we get a feel for what they're willing to pay a premium for, what attributes they find most valuable. So yeah, that one was a little wonky, but there are some that pop up that are more realistic.
So we know that consumers are willing to pay more if they understand what the seaweed is actually doing. And Miko, this brings us to the next part of your presentation, which is sharing some of those preliminary results on your project. So I'll go ahead and let you share your screen again. And Miko's going to bring us.
Can you see my screen?
Yep, we got you.
Perfect. Okay. Thank you. Very, very interesting. And yes, absolutely. I think that would be a nice, let me move the floating bar away. I really look forward to the questions section at the end, because I think that seeing all this, I was not aware of some of the things that it brings a lot of things together and answers a lot of the important questions that we have. So I definitely want to leave enough room for a little discussion towards the end. So this second part, I only have another 9 or 10 slides, I believe. So it's just basically dealing with our findings, and also want to touch on the challenges. And I want to expand on that, because that's really right now our bottleneck for that. But I know, let's start with, I know many of you wonder why, like, we're talking about bromoform and algae and seaweed. So this slide really puts it nicely. And I'm going back to, I recognize maybe in the audience, not everybody's familiar with why we're talking about all these things together: cattle, methane, and seaweed. So I wanted to just take 2 seconds to explain what's going on in the cow forestomach. So as you may or may not know, ruminants have different like fermentation chambers, rumen being the largest one, where there are a lot of different microorganisms. There's bacteria, protozoa, and then this other class of microorganisms. Oops, sorry, I forget that. They're called methanogens. So these are archaea, methanogenic archaea. They look a lot like bacteria, but they're not. And they are very specific individuals that they basically are in charge of getting rid of all this excess hydrogen that is formed every time cattle consume grass and any other feedstuff. It gets fermented. So the fermentation produces a lot of hydrogen and carbon dioxide. And then there is this methanogen that actually have a very important role in the cow's stomach. which is to convert or to get rid of all this hydrogen so that this fermentation process keeps happening. It's very important. So in other words, there's a reason why like methanogens exist in cattle, and it is to remove this hydrogen. So whenever we do, we have to make sure that if we come up with a way to reduce methane like we think we are, we need to make sure that we are not hurting productivity. And because there is a really good reason why methanogenic archaea exist and are there. So what is bromoform? Because you hear me talk already enough about bromoform, and also you heard Ashley talk about bromoform. So it's a very small volatile molecule that it is also in nature for a reason. It protects seaweed from predation. That was probably in the evolution of seaweeds. It's formed typically in vesicles. There's experts in this room that probably know a lot more about the metabolism of how bromoform it's formed and released to some extent from in microalgae or in seaweed. But in terms of cattle, we are learning, because we're relatively new to bromoform, that, as I'll show you in my next slide, has a key role in decreasing methane. And there's a lot of research out of Australia, that's where all of this started, where they basically identify, forgetting about this, Asparagopsis as one of the species. And you heard Ashley talk about red algae and brown. So Asparagopsis is considered red microalgae, whereas Gracilaria, which is the one that you will hear me talk a lot about, because that's the one that we have been harvesting with Aaron, and the one that now, at least we think that, and I'm not pretty sure that at least we got it right on the genus. I'm not sure about the species, but we definitely think that it is Gracilaria. We know that it's now growing in tanks, and I'll tell you a little bit more about that. The picture you see here is actually from those tanks when we went earlier this year to visit Two Docks facility, and we basically were trying. I'll come to why we're doing this in tanks later, but logistics probably the number one reason. If you guess logistics, you're right. We know that this bromoform can reduce methane. We are seeing that to a great extent. And the way it does, it inhibits some of the key enzymes, just without getting into a lot of detail. And the next slide infographic is going to do a great job of summarizing it. But basically interferes in the cycle of methane formation by this methanogenic archaea in the rumen, and basically creates a futile cycle where this archaea cannot produce methane as a way to obtain energy. And because instead of having a key enzyme in the process, this bromoform acts as an analog. So it is a safe feed additive. There's regulations. There's at least it's been identified. There's some provisions right now in the Food and Drug Administration to be able to feed bromoform with a special permission until it gets the full approval. But there's cattle today that have been fed bromoform and algae, I think, and I'm not 100% sure in all the regulation. That's been proving to be one of the most challenging parts of this project to me. It's just to find experts, that because a lot of people don't know. It's everything so new, especially at the regulatory agencies. Not one thing is bromoform, another thing is bromoform inside the algae. Another thing is a bromoform containing additive. So there's different ways to approach this, and to my knowledge, at least, just the feeding of microalgae to cattle is not. I don't think it has been maybe heavily regulated, but I'm sure there is some provisions that have to be in place. There's huge companies right now in Hawaii mainly that are producing microalgae, particularly from the Asparagopsis genus, to a large scale. So I'm sure that a lot of those regulatory issues have been contemplated. Right now, I can tell you for a fact that there is an opportunity to ask for permission for the Food and Drug Administration to temporarily feed to a certain number of animals, feed bromoform and harvest those cattle without any concern. So that is one of the areas that we're learning very fast. The next slide now that I'm showing here, it's the infographic that comes from this paper a couple of years back, does a great job of summarizing everything that is happening. First, I want to make one important point on this slide. Number one, just to disprove one of the myths that you see in the web, 95% of the methane comes out of the mouth, not out of the other end. So you'll see a lot of pictures sometimes or where there is this cow like with fire coming out of the rear end. That's entirely not true. Most of it, and then there's good reason for that. This large fermentation vessel is almost connected to the mouth. It's a much shorter via to whenever the cattle are eating, they eructate, they belch, and they release methane, whereas they have to travel through the entire gastrointestinal tract to be released on the other end. So it's a lot easier and convenient to release it on the front end. The microbes, I already introduced you to them, are bacteria, protozoa, fungi. There are some viruses, and within that there is the archaea that are the ones that produce methane. So fermentation in the rumen produces a lot of different carbon compounds, different volatile fatty acids that are a key source of energy for the animal. That's really these three here, acetate, propionate, and butyrate, are the main sources of energy. They are in charge of fueling the production of tissue, which turns into milk, beef, protein, etc. So leather. So all the production will be coming from these three main volatile fatty acids resulting from fermentation. And that's just as a curiosity, that's the numbers of which we find methanogens in the rumen. Typically it's 10 to the 5, 10 to 8 per ml of ruminal content. So this is just an idea, or for you to have an idea of how this whole thing gets pieced together. And one of the things that we're learning with Gracilaria is how much bromoform does it have. I show you early pictures where we were wild harvesting Gracilaria. So when we look at those early harvests of wild caught Gracilaria, we were looking at 17 to 34 nanograms per gram. And this is just to put it in context and keep in mind, we're talking nanogram versus milligram. We were talking about it order like 1,000 order of magnitude. So it's a huge amount in Asparagopsis compared to some of our early caught Gracilaria. But it's still a lot of room for improvement, and I will show you some numbers later that probably are a little bit more promising than that. It still contributes to methane reduction. So even it's a very linear relationship. The more bromoform, the more methane mitigation. So there is a reason why this multi-million-dollar investment in Hawaii it is done with Asparagopsis, probably because it's one of the microalgae that has the most bromoform. And again, remember, bromoform is produced by this microalgae as a defense mechanism. So there are ways to manipulate that. And in fact, as you'll see in one of my next slides, it might be the next one, that whenever we move away from wild caught into growing conditions in the tanks, we seem to do something that makes this macroalgae to have more bromoform, and that's very positive and very exciting. So the very early studies that we did were done again, going with Aaron trips into the Tampa Bay area, harvesting, trying to figure out ways to preserve that. This is some of the very early preservation methods that we had. We were just putting it in tanks, in bags and ice, and driving back to Mariana, which is close to 6 hours away from the Tampa Bay area. So it was quite a task. And I believe some of this last, at least I can confirm that I believe this very last Gracilaria, it is the one that is grown in tanks. So this is the latest. So since then, sorry again, we move away from the wild-caught gracilaria into, with the help of Aaron and with the help of the sea grant, really a portion of that sea grant was dedicated to the construction of these tanks where we can grow that in the Two Docks Shellfish facility. So now we're growing gracilaria. You can see the difference that we see, and we may know right now what species it is, but I think just being safe until we have 100% sure, we'll define it as gracilaria species. We have increased tremendously the amount of bromoform, the concentration of bromoform per gram of material. So I think one of the things that that you can see right now is the growing conditions being in tanks definitely seems to stress the plant. I'm not a seaweed expert or a phycologist, but the little I know is this stress tends to enhance the production of bromoform. Whether that's a reason or not, it has to do with nutrients in the tank. I'm not sure, but for whatever reason, moving from wild caught to growing in tanks has increased by a tremendous order of magnitude when you see those numbers, the amount of bromoform. So that's very exciting news. We were, needless to say, very excited on the first couple harvests we got from Aaron after growing this gracilaria in tanks. And this is just talking about minerals. I don't want to expand too much. It's a lot of numbers in this, but I try to make some distinction between all these are all analyses that we have sent to different labs to analyze for minerals. And why? Let me do a brief introduction of why we picked these minerals. There's some concern and very well-funded concern on levels of heavy metals, aluminum, arsenic, cadmium are some of the concerning. Calcium, copper, manganese, magnesium, manganese, phosphorus, and zinc are really good mineral for cattle. We actually supplement those. So I guess within some limits, the more of those, the better. So I'm not so much of a concern. But let's just spend 2 minutes on aluminum and arsenic. As you would expect, and that's one of the beauties of this organisms, they do pick up heavy metal from the environment, and they can be a way to clean our coastal areas and our waters. The bad part of that, if we're going to feed that to cattle, that could be a challenge. Luckily for us, when we analyze this contents, and without getting into a lot of specifics, none of these levels are concerning to the point of cattle toxicity. And of course, we have no data on tissue yet, or on how much of that translates into, let's say, beef or milk, but probably it's not a concern because it's not even concerning from a standpoint of the intake levels. I mean, of course, it all depends on how much of each of the seaweed cattle eat, but I'm thinking about realistically on what are the levels that would be needed of supplementation to create a product with lower carbon footprint. We may be talking about the range of 1 to 5% of the diet, and honestly, I think it might be leaning more towards the 1%. 5%, it's already a lot. So if we can feed something with that much bromoform included at a 1% of that diet, I think that might be part of that sweet spot that we're going to have to get back to talk to our economists and have studies to address the return on the investment. But I think that might be the inclusion level we might be discussing. So there are definitely the one that is cultivated in tanks has a lot less of this heavy metals. You can see how in some cases, first, a lot of variability between species. This first 3 were wild caught and transported and frozen and transported to the lab immediately. And then we started playing with the cultivated one. We play with different preservation methods. So we did what we call snap freezing. We took tanks of liquid nitrogen, took it to the Two Docks facility, immediately placed the algae in that, and then we brought it to the lab. You can see right away aluminum levels seem to be lesser in the cultivated. cadmium levels are definitely a lot less than what we've seen in the wild-caught species, or the same species, but different methods of cultivation. And the mineral, let's call it the good microminerals that would be for cattle, calcium, magnesium, they're a little variable, and they might have a lot to do with the media in which they're growing. So there is some of that, and phosphorus particularly. Take a look. That's one thing and that might be more of a question for the algae experts. Wild-caught gracilaria levels of phosphorus can compare with the levels, or the concentration of phosphorus we see in the wild caught. That's quite interesting. Sorry, in the cultivated, it's much, much more. So this is, again, from a cattle standpoint, is good. We do feed those minerals anyway, and they're not cheap. So I know I keep talking about nitrogen and methane mitigation, but I'm wondering if can you still hear me okay? Good. Okay. I'm wondering if we could perhaps enhance minerals in the algae and become not only a discussion about protein, but also a discussion about minerals, because they could be very expensive. So briefly touching on protein, because that's one of the main topics. So we also found some really interesting difference in how we're preserving the nitrogen in the algae. And this is really just to give you a reference in terms of cattle. If we were to feed some of this, 15% will be great, but probably will require feeding a lot of gracilaria snap frozen to meet the protein requirements. To put it in perspective, when I talk about cottonseed meal, we're talking about 38 to 40% protein. So definitely we have a long ways to go if we're going to supplement microalgae for protein. But I may be more excited after seeing some of the preliminary results about the mineral content, because we don't need a lot of those to create a lot of good in cattle, and definitely on the methane mitigation. As far as nitrogen, we might need to work with other approaches, like could be the microalgae. We actually do have a project with microalgae. We've seen some Nanochloropsis samples coming back to 30 to 40% protein. That's very exciting. So I see potential maybe in some of that on the protein side of things, but maybe not so much as a protein supplement, at least within gracilaria. So why is important to maintain bromoform level? Hopefully, it made enough of a case. It is the molecule that really has something to do with methane mitigation, and we think we know why now, but we do have to figure out ways to preserve it because it's very volatile. So one of the things that we've been experimenting with it is storage. So there's been some research out of Portugal that they've done some preservation in oils, stabilizing bromoform in oil and increasing the shelf life. Storage can cause a lot of problems instead of loss of activity of that. So the light seems to be an issue. So it's typically preserved in darker environment or in darker containers, so not to expose it to the light. And it's critical in order to have effectiveness in cattle. That is critical. So a lot of our research right now, it is concentrating on stability of bromoform. How can we make sure that the bromoform that we measure on the snap frozen microalgae that comes from Aaron's shop, it's preserved all the way to the time that we feed it to cattle. So one of the things we've been doing, and Aaron again has been helping us with that, realizing that the logistics of growing a lot of algae is going to be way more complicated. So we are now looking at ways to stabilize bromoform in oil. And that is something, again, that's been reported by researchers in Portugal. We've seen it a couple of years ago, the first report of that, mainly because of the lipophilic nature of bromoform. So it can migrate into the oil, and then so the idea would be at that time to concentrate the bromoform in the oil and then feed it to cattle. So that's a lot of the research we're doing right now. It's on that. So other preservation method we try, low temperature drying. It certainly does help. We've done freezing, snap freezing, just conventional. It works. It seems to help prevent the evaporation. It's like I say, it's extremely volatile. So freezing and particularly not freezing seems to work. I don't have a lot of experience with ensiling. Ensiling, it's the process basically like pickling, removing the oxygen, which certainly helps to prevent oxidation, but there's not a lot of research on that. And avoiding heat and exposure to air certainly helps. And I would throw into this slide also the light. Preserving it from exposure to light has a lot to do with how we're going to be successful. So,
Nico. Oh,
yes.
I'm sorry to interrupt. I just wanted to give you a heads up that we have a hard stop at 4:30, so we want to leave plenty of time for Q&A. So just kind of, you still have about 2 or 3 minutes. I just wanted to give you a
Okay,
time.
well, it's my last. I have only 2 more slides, so thank you for that. Yeah, I got carried away with my 10 slides, and it was 10 slides. I just checked. So I mentioned already the oil. So thank you for. Sorry. uh, um, um, I think I already mentioned that we're doing some work with that. It reduces moisture and it really helps us preserve bromoform. And this is the last take-home message. Basically, if I have to summarize the whole thing, we have identified some of those key compounds, mainly bromoform in Gracilaria. It is locally grown. We are able, and when I say we, here is Aaron, was able to grow it, and we just need to figure out a lot of details of the logistics. We now learning that we can preserve it in oil and maybe help concentrate. That would certainly reduce the amount of algae we need to create an impact on the cattle production. And this whole thing can lead to a very interesting bioeconomy in the coastal region, which is really, at the end of the day, the way that it was described at the beginning by Angela. That's really what we're after with that. So with that, I'm going to stop here, turn it over to you, Angela, to coordinate the questions. Apologize. I think we got maybe 11 minutes for questions if needed.
That was fantastic. Thank you so much. And thank you to all of our presenters today. I wanted to go ahead and take a moment to recognize Dr. Aaron Welch, who you've heard his name a million times, but you haven't seen his face yet. I'm going to ask Aaron, if you can, to turn on your camera. Dr. Aaron Welch is with Two Docks Shellfish Company. He is here in the Tampa Bay area. He's a shellfish farmer, but has recently been swooped into a lot of different seaweed cultivation projects, not only with the University of Florida, but also with Woods Hole, looking at tropical seaweed potential here in the state. So Aaron, do you want to go ahead and say hi, and then we can all kind of throw our questions your way as well?
Yep. Hey, everybody. Angela, thanks for putting this on. Nico and everybody else who's talked, thanks for the great presentations. Gabby, Angela, Ashley's not here. Great to listen to, great to be a part of it. And yeah, I'm here to take questions if you have any, just on the grower end. I'm not an expert yet, but we're working on it. And down the road, if you need to get in touch with me, Angela's got all my contact information. How about that? Good enough?
Yeah, well, the cool thing about this is, like, it's this intersection of Florida agriculture, right? We have our land-based agriculture, one of the oldest products in the state, which is Florida cattle, meeting our shellfish aquaculture industry, which is also part of ag. And, the shellfish aquaculture guys aren't producing seaweed, but they do definitely have seaweed aggregating on their shellfish leases. And this is sort of where the shellfish industry initially came into this conversation, as to like, hey, there's all this product that we're always continually removing from our shellfish bags. Is there anything that we can actually do with that product? So some of these different research avenues that are exploring potential uses and markets for these products, I think, could significantly benefit our aquaculture industry if anything is found out to be economically viable. So with that, I'm going to go ahead and open the floor for questions. You can type it into the chat, you can unmute, you can raise your hand, whatever you want to do, but let's go ahead and see if anyone has any questions. Go ahead, Raul. Hi.
Hi, hello. Thank you for this wonderful meeting and opportunity. I have a few questions, and for those who don't know me, I've been working in seaweed farming for 40 years in the Caribbean, and so I'm very much acquainted with experience in the region. And I would just want to bring up the two questions. One is, have you considered Euchematopsis siciformis? As you know, it's a very common species in Florida. It's highly domesticated, have a lot of different applications, and it grows very well. And I don't know about the bromoform content, but definitely it should a species that should be considered. And the other thing is that beyond the uses that you have for bromoform, and I know this meeting is concentrated on bromoform and cattle, but definitely there are other these species of Gracilaria you're talking about. Might be a Gracilariopsis, might be a Gracilaria tigvahea. I don't know if you're aware of the work done in Harbor Branch for decades, and I'm sure there is information that you can cross and find out more about it. But we in Venezuela, we are using Euchematopsis siciformis foscarragenin, and it's very successful, very good quality, and it grows very well. So I just wanted to know, why aren't you testing other species besides the Gracilaria? And there's any reason for that? And also, open to exchange information about it. Okay, thank you.
Yeah, I can jump in on that. We have tried Euchema. You said Euchema, right? Or Euchema?
Yeah, but now it's called Euchematopsis. It was changed for the Florida species. Yes.
Yep. So we have worked with that, and the short version is it just didn't work here. You're right; it's native to the area, and we found it wild a few times, but we could never get consistent performance from it, either in Tampa Bay or, in fact, Charlie Yarish is in this meeting with the University of Connecticut, and we had a project with Charlie and some folks at Woods Hole trying to do Euchema offshore out in the Gulf of Mexico on long lines, about 2 miles off the beach. Between that project and some attempts to grow it on our leases, which are inside Tampa Bay, we just could never get it to grow.
All right. And how about Asparagopsis? Have you ever tried? Because it's also available in Florida. It's actually available anywhere. It's a very common. Doesn't grow in large quantities, but
right.
It can grow in tanks. It can be grown, for indoor.
I would let one of the seaweed experts weigh in on that, but from our perspective, no, and it's largely due to what you just said. It's not common. We focused on the species that we see on our farm leases, which is primarily Gracilarias. Angela sort of mentioned it briefly. Every winter, starting about now until April, May, we get huge blooms of seaweed on the leases that have to be removed before they choke the clams or the oysters. And that's primarily Gracilaria. There's some other stuff in there, but mostly it's Gracilaria, and that's why we focused on it, just because it's here. But obviously, we'd be open to anything if it could be done.
Okay, thank you.
Yeah, thank you.
I have a question for Aaron. What's the biggest challenge of growing? Of course, I'm excited about growing Gracilaria, but what's the biggest hurdle? And I know we touched on that when we went there with you, but what would be your number one concern?
Yeah, I mean, I think you sort of touched on the two big issues for me. One is markets. There is no real market for anything that we've ever successfully grown. I mean, I know Gracilaria has a bunch of applications, agar, et cetera, maybe bromoform, but those markets are either scale markets, like mass production volume, you're competing with Indonesia and China or whoever, or they're for a product they're for an application that doesn't really exist yet. Like bromoform is exciting, but there's a lot of steps between here and there. For one thing, the FDA has to approve it. So there's not actually a market. And then the logistics. I mean, Nico, we're starting to figure some of these things out. Like, holy cow, we got to freeze this stuff the second it comes out of the water or it doesn't work. And that's no big deal when you've got 5 or 10 kilograms. When you've got 5,000 kilograms, you better have some stuff in place. You better have your logistics lined up and ready to go. And that's an enormous challenge. It's that way in any agriculture business, right? Like citrus producers, cattle producers, whatever, us, shellfish producers, you're geared up and ready to go. You've got your logistics set, but they're knowable and you understand them ahead of time. Right now, we just know that the logistical challenges would be huge. We're not quite sure how to solve them. So we don't, we just know it would be a lot. We're not quite sure what it looks like. So those two things, like the logistics and the markets, to me are the big issues.
Great, thank you. And on that, just to touch on that, that's the reason I agree 100%. I don't think freezing will be an option. That's why, based on research, we're doing some preliminary research here, and we've seen it in other countries. I think the option to maybe put it in any sort of oil, any cheap lipid, seems to work quite well. So we're going to have some results soon. That may be a way to solve that part of the logistic challenge, but the market still has to be developed. You're absolutely right.
Yeah, and I think the logistics with oil, I mean, we did that little trial for Liza a few weeks ago. That's even that, right? Like, it's going to be easier, but it's not going to be easy. You're still talking about lots of oil and having to move it in barrels and trucks and forklifts and etc. Suppliers who's going to sell you all the oil, and it would get thick pretty quick.
Are there any other questions out there for any of our presenters or for Aaron? Well, we have 2 minutes left. So before I let you go, I am going to request that you fill out a really quick poll. I'm going to launch it here in a second. It'll take you 2 seconds to answer the questions on it. And then maybe if anyone has any grand concluding thoughts, and then we'll close out right at 4:30. So I'm going to launch this poll right now. If you don't mind to go ahead and answer the questions. And if you're a presenter, you can participate too.
I think there was a question just asked about the fertilizer for grassland area. I don't know if Aaron is gone already, or I know he had to leave, but
Yeah, Aaron, are you still on? The
Yeah,
question
I'm
in the
still
chat
here.
from Charlie.
Yeah, I just responded to Charlie. It's Jack's liquid soluble. So what we have used is Jack's, I think it's 28.818 for the NPP content. I can double check on that, but it's definitely Jack's soluble. That was recommended to us by Loretta Roberson. It worked like a charm once we got them into the tanks. Not much, few micromole for 24 hours, 2 or 3 times a week, and that got it done.
Okay, well, pending no other questions, I will go ahead and close this out here. It's 4:30. Everyone who's participating in the call and everyone who's registered for the webinar, even if they're not here, will get an email with a link to this presentation, as well as just like a PDF with a lot of the references and resources that we've talked about during today's call. Feel free to reach out to any of us if you have any, future questions or clarifications or communication that you'd like to participate in. And I really thank all of you for your time and participation in this webinar. Thank you. Thank you to our research team.
Thank you. Excellent job, Angela.
Well, research
Thank
team,
you so
I'll go
much.
ahead and say goodbye.
Bye.
Yeah, thank you. I'll
you.
send the poll results to everybody in a bit. And thanks again for all the time.
Perfect. Thank you so much.
Thanks,
Bye.
Angela.
Thanks, Gabby.